All right, so we've established some of the signaling mechanisms in place that help entice us to eat, either as a part of stimulating appetite or hunger. And of course, at the opposite end, we talked about signals that stop us from eating. But once we have stimulated that sense of appetite, that sense of hunger, and we go and find food and put it into our bodies, what happens to it? And so next on that process, on that storyline, is digestion. We're going to need to break down these huge molecules that came from our food, such as the carbohydrates, lipids, and proteins. Macronutrients are really, really large molecules. And we need to break them down into their smallest pieces, parts, which are going to be called their monomers. We'll talk a lot more about monomers, which are the single smallest version of each of these nutrients of a type of molecule. And so again, our goal is to break down these foods into their smallest pieces, into their monomers, so that we can then get them across some cell membranes and into the body and to the cells that need them. And so we're going to focus on that breaking down part, digestion. So essentially what is happening all along your digestive tract or gastrointestinal tract. And as a reminder to some terms that were brought up much earlier in the class, as far as types of metabolism, digestion is a great example of catabolism, this word with a C here, which is referring to any chemical reaction in which a large molecule is being broken down into a smaller molecule. So this is going to release energy and, of course, release heat as a byproduct. Once those molecules have been broken down to small pieces, we will absorb them, and then they may be used to build up into other larger molecules, which would be the opposite, anabolism, right, with an A. So catabolism, breaking down, anabolism, building up, and all of digestion is essentially one large catabolic process. So your carbohydrates are all going to be broken down into monosaccharides. So back to that idea of monomers, the single smallest units of each of these molecules, the building blocks of these molecules. We want to break down all of these large sugars in our food into their monosaccharides. That would be one single sugar. So a simple sugar or your added sugars, those are typically monosaccharides. They're small, simple versions. Our lipids, we're going to break down into, there are two separate building blocks. Lipids are built up of glycerol heads and fatty acid tails, if you remember from our bit on cell bio. And so we want to try to break off, or we're going to break off as many fatty acid tails as we can, and typically leaving behind one fatty acid tail with that glycerol, so that would be called a monoglyceride. So breaking those fats down to those tiny pieces. And then our proteins, we want to get down to their individual amino acids or some small peptide, so a dipeptide or a tripeptide, which would be 2 or 3 amino acids together. But again, the goal is to split these down into their smallest pieces. And this is all going to be occurring within your gastrointestinal tract or GI tract, okay? AKA your digestive tract or your digestive system. This is what we're describing. So that gastrointestinal tract is the long tube from mouth to anus with various sacs along that tube that are separated by circular muscles called sphincters. So this really is one long continuous tube. If those sphincters were not there, if they were not contracted, if all of those muscles were relaxed in your gastrointestinal tract, this would be one long continuous tube with nothing blocking the way. So these sphincters, those circular muscles, essentially separate the organs of this GI tract into their different parts, and we're going to have slightly different digestive processes happening in each of those organs. And so the mouth is the first stop. We then swallow that food, and it'll travel down your esophagus, which is just a long tunnel, essentially, between your mouth and your stomach. Then the stomach will lead into the small intestine. The small intestine will lead into the large intestine. And then whatever you were unsuccessful at digesting and absorbing, whatever is left over, is going to be excreted from the body as poop through the rectum and anus. So the idea is that your food has all of this time throughout this entire tract from the mouth, esophagus, stomach, small intestine, and large intestine. All through that time, your body is going to be bombarding it with different chemicals and enzymes to try to break it down. And whatever it is successful in breaking down, it's going to try to absorb. But whatever it is unsuccessful in breaking down and absorbing is what we eliminate as poop, as feces. And like I said, along the way, each of these organs is separated by a special circular muscle called a sphincter. It's a circle like this. You can imagine a ring. And so essentially when it contracts, it completely closes up. And then when it relaxes, it opens. So it's like if you were to pinch a hose and then let it go, right? So water can flow through that hose as long as you're not pinching, but then if you pinch the hose, water is stopped. That's what's happening all along this tract. And this allows some control and separation, right? It would be, or would it be helpful for your food to be able to come back up your esophagus? Right? If you ever had like a little baby barf, you feel that little toxicity in your esophagus as you cough up a little bit of those gastric enzymes or maybe even some bile from the small intestine. That's not fun. We also have slightly different environments within each of these organs. Your stomach is very acidic, but then your small intestine is going to be pretty basic. It's going to be on the opposite side. And so keeping those environments separate is going to be important for the chemicals that are active in those different environments. So these sphincters are really helpful with separating the organs along this tube. And so you have a sphincter at both ends of each organ, right? At the top of your esophagus, so when you swallow, you're relaxing that sphincter so that food can go down. And at the bottom of the esophagus, regulating how much food is getting into the stomach and so on. And so beyond the organs that are directly part of that long tube, that are directly part of that GI tract, we have a few accessory organs that are going to be really important for secretions. So for adding chemicals or releasing hormones that are going to help with this process of digestion. And so we have some really basic accessory organs like your teeth and tongue, which clearly play a role in your chewing of your food before you swallow it. But then the rest of these are all related to those secretions and hormones. So releasing chemicals into the stomach, into the small intestine, and secreting hormones that are going to help regulate this process. And so we have salivary glands that are helping secrete saliva into the mouth. We have the liver, which is going to produce bile. Bile then gets stored in the gallbladder. The gallbladder releases that bile into the small intestine. And then we have the pancreas, which is going to release a lot of different secretions into the small intestine. So the pancreas is really, really important for producing a bunch of enzymes and this chemical called bicarbonate that is really important for your small intestine to be able to do its job as a part of digestion. So we'll talk about the role of these all along the way as well. Okay. So before we can finally actually get into digestion, I think the last topic here to address beforehand is the difference between mechanical digestion and chemical digestion. And so if you already have some association just with those terms, you probably have a pretty good idea of what they mean. Like mechanical, thinking like robots, mechanics, like the actual physical pieces of parts of a factory. And mechanical digestion is referring to the physical separation of things. And so this is not going to break actual bonds and releasing the potential energy that was stored in these molecules, but mechanical digestion is going to help start physically separating and interrupting some associations. There's a lot of like magnetic attraction that holds molecules together. So that would be an attraction rather than a bond. And so some of our mechanical digestion is going to be interrupting those attractions, putting a wedge in between that magnet so they can't get together. Right? And so some of these are more obvious, like chewing as an example of mechanical digestion, that physical force being added to your food to help break apart some of those interactions. And then some of them are a bit harder to understand, those weaker magnetic attractions, such as denaturing proteins, which come as these large, complex, folded structures that we're going to need to interrupt so that they can unravel, kind of like unfolding a pretzel into this linear structure, which we'll look at later. So mechanical digestion is physical separation and interruption. Chemical digestion is always going to be utilizing enzymes. So if you hear me talking about enzymes, or whenever we're asking about what an enzyme does throughout this process of digestion, that is referring to chemical digestion. Enzymes are specialized proteins that help break bonds or build bonds. They can go in both directions, but For the process of digestion, these digestive enzymes are all going to help break bonds. So anytime we're actually talking about breaking a bond holding atoms together, that is chemical digestion. So any reference to enzymes, that is chemical digestion. Right. One more concept before we move on, the idea of pH. So if you've heard of acids and bases, we don't need a super complex understanding for this chapter, but it is going to be referenced because again, we have slightly different environments in some of the organs along our GI tract. Your stomach is very acidic, right? Gastric juice has a very low pH, is a pretty strong acid. But then as that acidic gastric juice is moving from the stomach to the small intestine is going to make it more basic. It is going to be secreting bicarbonate from the pancreas and bile from the gallbladder, which are going to push it into this basic realm in pH, giving it a pH of around 8. And then your saliva, what's in our mouths, is close to neutral, a neutral pH of 7, almost like water. So along the way, we are going to need to track this pH adjustment to understand what those environments look like, because that is going to allow for some enzymes to be active and other enzymes to not. pH can turn on and turn off different chemicals, and so that's what's happening along the way. All right. So now getting into digestion. We briefly introduced in the ingestion part of this chapter the idea of preparation, this cephalic phase, that as you are about to consume food, your body prepares. It is going to tell your salivary glands to start secreting saliva and tell your stomach to start preparing its gastric juices, secreting hydrochloric acid and some of its gastric enzymes. And then our gastric phase is what is happening once food is actually getting there. So all these regulatory mechanisms like hormones and nervous signals that are going to help with digestion, as well as telling your body that you are full, you have food. So this is all kind of repeat from the last portion of the chapter, right? As you are thinking about food or smelling it, or your body is reacting to lack of food in the stomach or low blood sugar, those signals are all going to be integrated by the medulla oblongata of your brainstem, which is then going to send out a nervous signal to tell your salivary glands and your stomach to start preparing, right? That cephalic phase. And so now that you have saliva and your stomach is starting to secrete its enzymes, we can now bring food into our mouth and begin the process of digestion. So digestion starts right away with food in your mouth. But the obvious one, chewing, producing some mechanical digestion, that physical force of breaking down these large bites of food into some slightly smaller pieces so that they are small enough that you can swallow them down your esophagus to your stomach. So chewing is a big part of mechanical digestion here. Saliva, technically, we consider part of our mechanical digestion, as it is softening and lubricating that food, making it more of a semi-solid mush to help with that movement down the esophagus to the stomach. But we do have a little bit of chemical digestion happening in the mouth as well. So we mainly are going to consider carbohydrates here. There is a bit of amylase, the enzyme that helps us chemically break down carbohydrates. There is some salivary amylase in your saliva. So the chemical digestion of carbohydrates does kind of begin in the mouth. If you think about like putting sugar in your mouth and just holding it there, it kind of seems to dissolve and dissipate into like a sugary paste with your saliva, right? That's because of this chemical digestion from the amylase in your saliva. We do technically have a little bit of lipase, which is the enzyme that digests lipids, in your saliva as well, but it's very minimal. So we're really just going to consider chemical digestion of carbohydrates in the mouth. So for this first part of your GI tract, for this first step in digestion, we have mechanical digestion of everything through chewing and through saliva. And then we have chemical digestion of really just carbohydrates, just some salivary amylase. So once food is ready to be swallowed, it just moves down your esophagus. There is no digestion or absorption occurring in your esophagus. It is basically just a tube to move past all of your other organs within your rib cage to get down to the stomach. So no digestion happening in the esophagus. But then we will get to the stomach. And the stomach has a lot going on. There's a lot that we could potentially expand upon and dive into and talk a whole lot about. I try to keep this brief, much briefer than what's talked about in the textbook. But some important ideas to point out here. The stomach is highly folded, and the small intestine and large intestine both have folds as well, and even more so. But as our first stop here, the stomach has a bunch of folds in its tissue, and that allows for it to expand and contract, right? To stretch as food enters and fills that space, and then contract as food leaves. This also helps with surface area for how much of these cells that are lining the stomach are interacting with the food that's being digested. And we have a lot of different types of cells that are lining the inside of your stomach. So we have mucus cells, which, as the name suggests, they secrete mucus, which is really important in your stomach because the gastric juice in your stomach is very acidic, which would be damaging to the rest of your cells. So that mucus is really helpful in coating this lining and protecting it from that acid. We have parietal cells, which secrete hydrochloric acid and intrinsic factor. Intrinsic factor is a molecule that's not really going to come up again for a while in this class. That really just helps with the absorption of vitamin B12, which is a unique little vitamin to talk about, has its own molecule for absorption. But again, that'll come up later. But more importantly, these parietal cells secrete hydrochloric acid, which is a super critical part of your stomach's role in digestion. As we said, the stomach is acidic. That's coming from this hydrochloric acid. Hydrochloric acid makes the stomach juice acidic, which is really important for activating certain enzymes that I'm going to talk about in a second, and killing off invaders, bacterial, fungal, maybe even viral invaders that came in with your food. A lot of those are going to be killed off just by simply being exposed to the acid in your stomach. That's a great benefit of this hydrochloric acid. Its secondary important role is activating the enzyme pepsinogen. So our next cell on this list are chief cells. They secrete an inactive version of this enzyme, pepsinogen. So pepsinogen cannot do its job. It is inactive. But when pepsinogen interacts with acid, when it interacts with hydrochloric acid, that is going to turn it into the active form, pepsin, which is now going to act as an enzyme to chemically digest proteins. And I guess that brings up another important role of hydrochloric acid that I kind of skipped over there. That acid is also going to help with mechanically digesting protein. And I'll talk more about that in a slide later. And so lastly, we have endocrine cells. Any endocrine cell, any place in your body is responsible for releasing hormones. And so the endocrine cells of your stomach secrete the regulatory hormone gastrin, which helps with this positive feedback loop of foods in the stomach. We release gastrin. Gastrin helps the stomach release even more secretions to help digest it. So this slide is to just help break down what all of those secretions do so that you don't necessarily need to write it all down just from me talking about it. Okay, so make sure to spend some time here taking some notes on what these different secretions do. Okay. One of the unique parts of our stomach is these multi-directional layers of smooth muscle. So I've talked a bit about stomach contraction and relaxation as a part of stimulating hunger versus satiety. This is also going to be helpful in mechanically digesting our food. Your stomach has muscles in all sorts of directions, so it can contract in all sorts of directions, which helps kind of mix it up, shake it, like a protein shake in a shaker bottle, helping that protein powder dissolve into your water or milk, right? As you shake it up, it just helps, or if you think of anything shaking, I guess, it helps it mix. That's what your stomach is doing all on its own. It is contracting all these different muscles in different directions, helping mix your food with all of those secretions to help with this process of digestion. So again, we can separate by what mechanical and what chemical digestion is happening in the stomach. So mechanical digestion, we're getting from that mixing, that physical force, pushing these molecules around and exposing them to the different chemicals. The technical term for all of what's in your stomach once food mixes with a hydrochloric acid is called chyme. So if you ever hear the term chyme, that's referring to food and all these gastric secretions from the stomach. So highly acidic, right, from that hydrochloric acid. Hydrochloric acid is going to begin the mechanical digestion of proteins. So it is going to start denaturing, which means it like helps unravel proteins. Proteins have a very complex folded structure. Hydrochloric acid starts interrupting those magnetic attractions that hold that structure together. And so going from a complex interwoven pretzel-like structure, starting to unfold into a more linear structure, that's going to be really important for the enzyme pepsin to then get in there and start cutting bonds and breaking apart amino acids so that we can absorb it. So hydrochloric acid denatures proteins, that is mechanical digestion of proteins, and it is going to help activate pepsin so that pepsin can chemically digest those proteins, can start breaking the bonds holding amino acids together. So this slide is just show you what I'm talking about with that complex folded structure of proteins. So the proteins that are being utilized in a cell are either going to be in this tertiary or quaternary structure, these images that we have at the bottom here. So lots of folds, very complex, and this is being held together by bonds that can be interrupted by things like exposure to acid. And so hydrochloric acid is going to start interrupting these attractions and bonds and unfold it into secondary all the way back to this primary, this basic linear structure. And once we're in this linear structure, we can now actually get in between each of these amino acids and break these bonds. While it's in this folded structure, these bonds are all protected. You can't get, pepsin is unable to get in there and break bonds, or at least it's not able to break very many of them. And so hydrochloric acid helping to unfold and straighten out this protein is really helpful so that these enzymes can actually break those bonds, chemically digest them. And so a couple of the major mechanisms regulating digestion within your stomach are related to those mechanoreceptors that respond to stretch. So as food gets to the stomach and those stomach muscles are stretching, mechanoreceptors sense that stretch, which is going to result in more hydrochloric acid and more pepsinogen secreting, right? Kind of great. Your stomach senses food is here. Okay, I can secrete even more of the enzymes and juice that I need to break down that food. And then those peptides that are being broken down, those small chains of amino acids, after pepsin is starting to break some chemical bonds, those peptides are going to bond with peptide receptors along the stomach. And that is also going to help with the release of more hydrochloric acid in stomach muscle contractions. So we have a lot of what would be considered a positive feedback loop. We have a lot of positive feedback loops in our stomach. So as food is entering and being digested, that is going to tell your stomach to do even more, release even more enzymes, more juices, more digestion. So a very well-maintained system to help digest and continue digesting your food. So then from the stomach, we move into the small intestine, which is going to play the largest role in both digestion and absorption. We're going to get chemical digestion of everything here and absorption of basically everything here, which we'll talk about in the next part of the chapter. Okay. But so as your chyme, that mixture of digested food and all of these gastric secretions in your stomach is leaving the stomach, it's very acidic. And so as that enters the small intestine, one of the first things that your small intestine wants to do is buffer that acidic chyme and make it more neutral or even basic. Because again, that acid is not only dangerous to the invaders that might be in our food, but it would also be damaging to your cells. And that's why the stomach has mucus cells to help protect it. Your small intestine has some mucus cells too, but it's going to try to completely take away that acidity so that it is now safe to travel through the rest of your digestive system. And that is also going to allow for all different types of enzymes to be active once we get rid of that acid. So I guess also on this slide, something to point out, these two accessory organs that we briefly introduced earlier, the pancreas and the gallbladder, directly secrete into the small intestine. You can see if you follow the green of the gallbladder and the yellow of the pancreas, they have these connections that are going to allow them to secrete things into the small intestine along with our food. These are going to be really important for digestion here. So We have our pancreas, the gallbladder, and then the liver just produces the bile that gets stored in the gallbladder. So let's talk about all these things here. So we have our very acidic chyme coming from the stomach into the small intestine. The pancreas produces this chemical called bicarbonate, which is essentially just a base that is going to help neutralize or buffer that acid. So this is one of the first secretions that is going to be released into the small intestine, this bicarbonate, to help take away that acidity and make it more basic. We then have bile from the liver. So the liver produces bile, stores it in the gallbladder, and then the gallbladder secretes that bile into the small intestine. And bile is a really cool molecule that is essentially a special lipid, a special type of fat that helps break down your fat into smaller pieces. And so that is a process called emulsification. So we have special names for mechanical digestion of fat and mechanical digestion of proteins, I guess, right? We said denaturing when describing mechanical digestion of protein. Emulsification is describing the mechanical digestion of fat here. That's going to help take the big globs of fat that come in our food and break them off into smaller little blobs and globs to help with digestion. So we'll take a look at more of that in a minute here. And then we have chemical digestion of all of our macronutrients within our small intestine. So the pancreas, this specialized accessory organ here, produces all types of enzymes. We have proteases, lipases, nucleases, and amylases. And a pattern you may have picked up on here is that suffix ase is always referring to an enzyme. So lipase is referring to an enzyme that breaks down lipids. Okay? Lactase is the enzyme responsible for breaking down lactose. So if you are lactose intolerant, that's because your body is no longer producing the enzyme lactase, or it never had the ability to produce the enzyme lactase. Okay? So if you see the suffix ase, A-S-E, it's referring to an enzyme. So proteases are enzymes for breaking down proteins. Lipases are enzymes for breaking down lipids. Nucleases are enzymes for breaking down nucleic acids, which we don't talk a whole lot about in this class, but that's what makes up DNA. So you're eating living things, so you are going to be breaking down DNA and RNA. And then amylase, referring to the type of carbohydrate called amylose, is the enzyme that breaks down carbohydrates. So we're mainly concerned with proteases, lipases, and amylases, these enzymes that break down our macronutrients. And these are all present in the small intestine, thanks to the pancreas. All right. So going back to bile and this special mechanical digestion of fat. Because fat is hydrophobic, it tends to clump up together. Right? The stomach is a very watery environment with all these different secretions in it. So all the fat from your food is going to form these large droplets, these lipid droplets, which are big clumps of fat. And that's going to be protecting all the fat that is stored in this big glob. And so if we want to be able to chemically digest it, expose it to lipase, we have to start separating little pieces. And that's a process called emulsification. And that is carried out by the molecule bile, again, produced by the liver, secreted by the gallbladder. But bile are these specialized lipids with salt on them, which allows them to have a little hydrophilic region to them that can interact with water. Those little lipids are going to go and kind of pull off these much smaller little chunks of fat, and they're going to help the enzyme lipase get in there and start digesting it. And so lipase will go in and chemically digest those lipids, and then you're left behind with a structure called a micelle that looks like this. It's a tiny single layer of lipids that can just easily diffuse into the cells that line your small intestine. So it's kind of cool, this process of taking smaller little clumps, letting lipase come in and chemically digest it, and then you get this nice little bubble, this little droplet of digested fats that can just go and easily diffuse into the cells that are going to absorb them. And so the chemical digestion, what lipase is doing, is separating fatty acid tails from these glycerol heads. And you can try to ignore, for right now, all of the elements that are being shown. I don't need you to recognize the carbon chain and actually know the element difference between a glycerol and a fatty acid tail. But I included this more to just show that these fatty acid tails can all be separated from their glycerol head. So the fat you're consuming is mostly coming as a triglyceride. It looks like this. And we need lipase to get in there and start breaking off some of those fatty acids, giving us smaller molecules that are much easier to absorb and get into the body. And we have a good bit of control mechanisms that play a role here in the small intestine in regulating your digestion. So these are all going to be chemoreceptors this time, whereas we had both mechano and chemoreceptors in the stomach. Some of the control mechanisms I would want you to be aware of are the response to acidity. So as soon as that chyme moves into the small intestine, that is going to stimulate secretin, this hormone that goes and tells the pancreas to release bicarbonate. So in response to acid, the cells in the small intestine secrete the hormone secretin, and secretin tells the pancreas release bicarbonate so that we can buffer that, we can neutralize it. And then as food is being chemically digested, your fats and proteins are going to start bonding with the receptors that are going to bring them into the body, that are going to absorb them. And that results in the secretion of enzymes like CCK, which produce this positive feedback loop for the small intestine. They help the gallbladder release more bile. They help the pancreas release enzymes, but it's going to have a negative feedback loop on the stomach. So as food is getting into the small intestine, we don't need to keep secreting those gastric juices and enzymes into the stomach. Theoretically, or the idea would be that all of the digestion that was going to happen in the stomach has already finished. So CCK and secretin actually have a negative feedback loop with the stomach. They tell it to stop secreting its juices and to slow down and no longer need to mix. So as food leaves the stomach, hormones are going to be released that tell the pancreas and gallbladder to release their secretions. And those same hormones are also going to tell the stomach that its job is done and it can stop. So pretty cool interwoven system. And so hopefully, by the time food has finished in the small intestine, you have successfully digested everything that you were going to digest. So all the mechanical and chemical digestion that is being accomplished by your body's cells should essentially be done by the time whatever is left is getting into the large intestine. So typically, whatever is left by the time it's getting there, it should just be mostly fiber, a type of carbohydrate that you cannot chemically digest. Water, because some water, maybe some minerals and vitamins might still be sticking around. And then bacteria that was in your food. And that bacteria is going to join with the trillions of bacteria that already live in your large intestine. So your large intestine, aka your colon, is home to a majority of your microbiome, or all of your gut microbiome, all the bacteria that help your gut. And these are beneficial. You have lots of probiotics, beneficial bacteria living in your body that are going to help with some digestion that you were incapable of, such as digesting fiber. So some of the fiber in your diet is not only just there to help you feel full and help clean up the lining of your intestinal system, to help with that gastrointestinal health, but some of that fiber is actually going to be digested. It's just being digested by a different organism. It's being digested by the bacteria that lives inside your large intestine. And as a result of that, the bacteria are going to release some small fats, some short chain fatty acids that you can then absorb and utilize as well. And so whatever made it through your entire digestive process to your large intestine is then going to be stored there for around 12 to 24 hours until it is finally excreted as feces, as your poop. So the more molecules in your food or parts of your food that are undigestible and unabsorbable, the more stuff you're going to have stored in your large intestine for 12 to 24 hours. And thus the more lethargic and slow and full and weird your stomach feels, or not your stomach, but your gut feels, because you have all this undigested stuff sitting there in storage. So if you are mostly eating real food that is all digestible and absorbable, there's very little for your large intestine to store and deal with. And so it's a lot more regulated. And so that basically it. We have a couple summary slides here at the end for you to look at and take reference to. But yeah, that is the entire process of digestion. So to kind of quickly summarize, we do kind of, this is a great slide for it. So in the mouth, you have mechanical digestion of everything from chewing and from the lubrication in your saliva. But we really only have chemical digestion of carbohydrates from salivary amylase. So that food then moves down your esophagus, where there's no digestion happening in the esophagus, but then gets to the stomach. In the stomach, hydrochloric acid is going to help denature, mechanically digest proteins. And the churning of your stomach with stomach muscles is going to help mechanically digest everything, exposing it to those molecules. And then we really only have chemical digestion of proteins in the stomach. So the enzyme pepsinogen becomes activated into pepsin by hydrochloric acid, and that enzyme is helpful for digesting proteins, so chemically digesting proteins. So from then, your food moves into your small intestine, where it is going to be exposed to all different types of enzymes. So we have chemical digestion of all macronutrients in the small intestine, and we have mechanical digestion of just fat, of just lipids from that molecule bile that helps emulsify large clumps of fat into smaller clumps of fat. And then the idea would be that by the end of your small intestine, you have digested all of these macronutrients in your food. And so all that's left moving into your large intestine is going to be fluids, some minerals, and then fiber, right? That carbohydrate that you cannot digest on your own. Okay. So that is all for digestion, and I will see you in the final part of chapter 3 on absorption.